Okay, it's 9 o'clock and we are ready for the 24th class in the UBC Ecosystem Modeling course called Fish 501. And I would like to welcome you. And today we have Marta Coll from Barcelona giving a talk, and I will get back to that in a few slides. But just notice that this is where she works when she, at least it was where she worked when she was able to go to work. The CSIC, the institute, is placed down there underneath that hat that looks like a fish here at the end of the beach next to the Olympic Harbor in Barcelona. But I would start though by acknowledging here that we here at UBC are in Coast Sally's territory. And as such, we acknowledge that we are at the traditional, ancestral, and unceded territories of the Musqueam people, the Tsleil-Waututh people, and the Squamish people. Also, once again, reminding you that the recordings from Fish 501 are available at the Ecopath YouTube channel and the Facebook page, and that you can find the lectures, tutorials, and other things at the course website, which is at a new Google site. You have the address here, so I encourage you to join and check that out. And also mentioning that we have a Slack workspace, which you can get invitation to join if you send me an email. And with that, I would like to welcome Marta. Marta is a dear friend and colleague, and we've been working together for a very long time. It's almost 20 years, Marta. And she has an enormously wide repertoire in what she's doing. Ecosystem modeling, of course, statistical methods, looking, evaluating biodiversity, some fascinating work on that, and an incredible high productivity. She's also central to the development of EWE and is a part of the not-for-profit research organization called Ecopath International Initiative that is coordinating the development. And I think, well, no, I don't think, I know that she's going to be talking about development of the global ocean model and how it's being used. And I'm very pleased to welcome Marta.
Okay, so it's great to be here today. Thank you, Billy, for the nice presentation, for showing that I'm not that good at paddling yet, although I'm trying. And yeah, it's great to be here today to present some of the work that we have been doing, going global with the application. Well, Billy already introduced me. I'm from the Institute of Marine Science in Barcelona. This institute is one of the larger institutes in the south of Europe and the largest marine institute in Spain. And basically, we have been developing research in three main challenges that you have here: understanding ocean and climate interactions, conservation and sustainable use of marine life and ecosystems, and the comprehension and mitigation of anthropogenic and natural hazards. And basically, my group intersects these three challenges in many of the work that you have been doing. And we work quite a lot with models, and we work quite a lot with local and regional models. But today I'm not going to talk to you about this local or regional work, but about EcoOcean, which is an attempt to go global with a trophodynamic model. Before, let me introduce you the EcoOcean core team that you can see here. These are the people that have been basically behind the latest developments, and obviously Billy from the beginning, then Jeroen and Joe as developers, as some of the programmers of the model, Maria Gracia Penino, a colleague from EO, also helping with some of the post analysis, and myself. But also this kind of big endeavors are include many other collaborators like Dr. Walters and our colleagues from Aquamaps, Reg Watson, etc. So to all of them, I would like to acknowledge, and this is really a collective effort what I'm going to present today. So why to go global with models? And this is something that it's quite obvious when you look around at the challenges that we're facing today, that human impacts are spread globally. We have climate change that impacts marine ecosystems worldwide, but also there's teleconnections between what's happening in one ocean base that at the end ends up impacting what happens in another ocean base. So there's really the need to integrate some of the analysis that we would do. Also, the anthropogenic impacts affect marine biodiversity widely. And there has been actually for quite a long time large efforts from the scientific community to do global analysis. I think the most obvious analysis, the most obvious initiatives are the IPCC work that has been releasing periodical reports for a while, and also the IBBS reports, right? The Intergovernmental Science Policy Platform on Biodiversity and Services. Just wanted to quickly showcase some of these reports. For example, here you have an infographic that shows some of the results of the last the special report on the ocean and cryosphere in the changing climate, where IPCC documented very interesting historical but also future projections of changes in ocean variables globally. And you can see here, for example, temperature, pH, caloric content of the ocean, the permafrost, the surface, etc. And all of them showing quite worrisome trends. Also, we have the efforts of IPBS that I mentioned before, where they really go beyond the assessment of climate change to other direct drivers, including land sea use changes you have seen here, direct exploitation, pollution, linking it with indirect drivers, and also assessing not only in the ocean, but also in the freshwater and in terrestrial ecosystems, what is the impact that humans are having in nature, and projecting as well what the impact may be. So it was, I think, natural for us to transition to try to go also global and do something global in a trophodynamic model. We are not the only ones working with ecological models at the global scale. There's several attempts that I'm going to introduce as well today. And there's models that, for example, use size distribution-based principles or size-based principles, composite models, etc. And because of this increasing computer power, big data availability, and the modularity of Ecopath with Ecosim that you have been shown these days, we decided, or basically Billy decided to do this attempt to build this trophodynamic global model. And this is how all started with a first attempt that was published in 2015, where he published a paper that is called The Global Ocean is an Ecosystem. As I told you, we are not the only ones. There are several groups and several colleagues that are also working in developing global marine ecosystem or marine ecological models. And I'm going to introduce here the Fish-MIP initiative, because it's something that I'm going to be referring quite a lot in my presentation today. This is an initiative where basically brought the community together that were either developing global models, but also regional models, and brought us together to work and in the standard formats on how we drive our models and how we compare the results of our models. It has been a great community learning experience for many of us, and I'm going to refer to it later as well. And obviously, this Fish-MIP initiative feeds also to PBS and IPCC reports sometimes. Here you have a little bit and the historic evolution of EcoOcean, the precursors that were the work that were started by Jackie Alder and Billy, trying to model larger areas of the ocean in a standardized way. So here you have the first word of the 18 global fishing areas of FAO, and also the 16 large marine ecosystems of Billy in 2009. That brought in 2015 to the first EcoOcean version 1. And some years later, basically, with this model, we developed it a little bit more, and we were able to participate in Fish-MIP, as you see here. In the Fish-MIP initiative, we have been using EcoOcean for several analyses. We have modified it, and we released last year another version, which is called version 2, that I'll introduce today as well, where we are hoping to contribute to new iterations of Fish-MIP and release probably next year a new version 3. Okay, so what is EcoOcean? And especially in version 1. EcoOcean, as I said, is a trophodynamic-based modeling complex. So it's not a model, it's really a linkage of models that try to understand the distribution or try to predict the distribution of species at the global level. And it was developed with the first aim to evaluate the impact of management actions and seafood within the ecosystem. It is spatially resolved and can run at 1 degree or half degree. It integrates the water column, so it's a 2D model, and frequently or by default, it runs in monthly units of time. It basically distributes or aggregates the biodiversity of the ocean in functional groups or species groupings that we call. We have around 50 that are linked in a food web, and we include from bacteria all the way to seabirds and whales. Of course, we also include the fishing fleets. We have 14 fleets. And we also the functional groups are divided in sizes. So we have small organisms, medium organisms, and large organisms for the fish especially. It was the first parameterized to represent 1950 in a similar way as we do an Ecopath model, and then we fit it to time series. And it's validated with global fishing statistics. And what is relevant today is that at the core of the computation of EcoOcean, it's the ecospace model that you have been introduced in this course. I'm not going to extend today about what is Ecopath with Ecosim and Ecospace, because by now you all have been listening to this quite a lot. But just to remind you that we have a core, the Ecopath model, where it gives you basically the snapshot of the structure and the flows within the food web, and describes the balance between how much is produced and how much is consumed in the system. And later we have these temporal spatial model components that link to the parameters of Ecopath, and we have the temporal one that simulates the ecosystem effects of mortality changes because of predation or because of fishing, and environmental forcing over time. And after that, you have the ecospace, which is the spatial temporal model that, for example, allows you to predict the spatial dynamics in a 2D space. Okay, so basically, EcoOcean does is extends the modularity of the EWE approach, especially Ecospace, which is this ability or this capability to work with other models and to work with other data. And it takes a big advantage of the spatial temporal framework. So you can drive models with dynamic layers to connect to a GIS environment, as Jeroen Stenbeck presented to you. And it also takes a big advantage on the habitat foraging capacity model that you have also been introduced, basically interlinking the capacity of the food web within the niche modeling environment. So this here you have the structure of the initial version of EcoOcean. EcoOcean, as I'm explaining you here, has this static core where we parameterize the initial conditions as an Ecopath model, where we bring in data from other models, from global databases. For example, you have here Fish-base. Then there is a temporal part of the model where we run the model with a customized version of Ecosim, and we have, in this case, fitted it to time series of historical global fisheries. And then we have the spatial temporal domain of the model, where all the spatial analysis happen. Here you can see, for example, there's the food web or the ecospace core that obviously is driven by the habitat capacity model, and the habitat capacity model is either driven or forced with data from Earth system models that I'm going to introduce to you later through the habitat capacity and the response functions, or directly through, for example, changing layers like the primary production of the base of the food web. At the same time, we have fish-based or aqua maps feeding, or other types of models. You could have other types of models feeding environmental preference functions and then go into driving this habitat capacity model. Okay, so you can see that it's quite flexible, and also there's quite a lot of interlinkages between databases, parameters, and model components in this complex. We also rely in the gravity model that has been introduced before by Billy to specialize the fishing effort that comes from the initial conditions from the time series, and then goes spatial with a customized version of EcoSpace, and we also allocate by LME. I'm also going to show you this later. So what are the drivers that we use to drive EcoOcean? We have depth, obviously, and here in this map you can see the depth layer that goes into EcoOcean when we run it with 1 degree. You can see there's around 43,000 cells in this layer. If we run it a half degree, the number of cells is 171,000. So you can imagine the capacity, the power capacity, the computer capacity that you need to run one of these simulations with EcoOcean. We also have a series of phytoplankton concentrations, or the base of the food web, that are forced with historical data from Earth system models that I'm going to show you later, and are also with forecast data from these models. And then we have a series of environmental layers. EcoOcean version 1 was basically driven with temperature. This was surface temperature, depth integrated temperature, but we are also now including other drivers. So we can include oxygen, ice cover, salinity, and how it changes at the global level. And there's the historical fishing effort of 14 fleets. These figures come from the original paper in 2015, where this was a first attempt to compare the historical fitting or the trajectories, the projected trajectories of the fishing in the historical period from EcoOcean, what we had at the global scale. And the fitting was pretty good. You can see here in the first, the two figures on the top, A and B. These are the matches or the comparisons of the catches between observed catches per functional group and the predicted catches of functional of the species groupings of functional groups in the model. The C and D are the catches or the comparison by LME, so by large marine ecosystem. And here you can see that we did a bit worse in matching all of the regions. So there's actually quite a lot of work to be done in this direction, and we are hoping to move in new analysis of how we are going to fit the model to, for example, regional averages in the near future. Still hear me well? Yeah, great. Okay, so EcoOcean version 1 has been, as I told you, we participated in the Fish-MIP initiative. It was a great opportunity for us to test our model complex and develop scenarios of climate change, because Fish-MIP allow us to get data from Earth system models, so climate, geochemical models that give you a series of environmental parameters from global models like temperatures. You have here in this figure to the left, you have sea surface temperature, net primary production, and phytoplankton concentrations. Here you are seeing two of the Earth system models that were available under CMI P5 in Fish-MIP. One is the GFDL model, and the other one is the IPSL model. I'm showing you this because later you're going to see the impact of using these two types of models in EcoOcean. And here you can see, for example, in the figure, how these two Earth system models predict some different patterns in terms of the net primary production or sea surface temperature for the ocean. In the figures on the top, the first two, you have the I think it's 1990, 1999 net primary productions for GFDL and for EPSL. So you can see that you already have some differences these alternative Earth system models produce. And later in the other four figures on the bottom, you see their projections of this net primary production change under two contrasting scenarios of or RCPs, so contrasting scenarios of emissions. You have 2.6, which is quite an optimistic scenario of emissions. So it means that temperature is predicted not to increase as much as under RCP 8.5, which is an extreme scenario of warming, as you can see here also on the time series. The same figure, but for temperature, you have it in these other maps, where you see the warming, the patterns of temperature on the top, and then you see how the projections of warming, if you're using either one of the two Earth system models, GFDL being much more or less pessimistic about warming than EPSL being more pessimistic about warming. Sorry, and here this was the net primary production. So I got confused. This is the figure of the net primary production. So in blue, the areas where those models are projecting an increase in primary production in the future. In red, the areas where those models are projecting a decline in primary production. And here, the areas where the models are projecting an increase in temperature. You can see that basically most of the models are obviously projecting an increase everywhere in terms of temperature, but there is an important contrast between what it's EPSL, RCP 8.5 projecting, or what is GFDL 2.6 projection. So there's quite a variability in what's projected to happen in terms of these environmental variables. And I am talking about these two contrasting models. Here you can see probably better what I mean in the figure to the left. This is an ensemble modeling comparison using the models, all the models that you have in the right. So you have a series of models with their projections, and in the left you basically have their mean and the spread of what the models are projecting for the 4 RCP scenarios. So taking into account all the models that you are seeing in the right, and if you look to the left, you see that for RCP 8.5, most of the models are projecting a higher increase on temperature, while if you look at RCP 2.6, the models are projecting a less increase in temperature. What we did in Fish-MIP, and we have followed in other exercises of EcoOcean, is we kept those contrasting Earth system models as our alternative scenarios of environmental conditions. So when you see here, the blue dot is EPSL model that we are going to use also later, where you see this is a model that predicts larger increases in temperature, while GFDL would predict under the same scenario of warming, RCP 8.5, lower increases in temperature. The same thing you can see here for the net primary production, where GFDL, which is the red dot under an RCP 8.5, predicts slight declines of primary production in the ocean, is not as extreme as what EPSL is predicting, the blue dot, which is really predicting important declines for the 2100. Okay, this is important because these are the drivers that later we are going to be using for our trophodynamic model, our model of the global ocean, adding or using these layers as drivers. So whatever we use, it's important later on, you will see. In this specific first study for Fish-MIP, we not only looked at standardized outputs of the Earth system models that we could use for our marine ecosystem models or marine ecological models, but we also looked at how we could standardize the results, how we could compare the results from all these models that we were running at the global level. In this table here on the top to the left, you can see a list of all the indicators that all the models that were participating at that time in Fish-MIP could produce. We have things like, for example, total system biomass or total consumers biomass, biomass of organisms that are larger than 10 centimeters or larger than 30 centimeters. That was the way for us to all compute the same indicators where we were running our models. On the left bottom, you see the results that EcoOcean, using version 1, was producing for total consumer biomass under these two contrasting Earth system models, so using GFDL and EPSL, as I showed you before, and also using the four RCPs, the four contrasting RCPs. So you can see the quite different or variability in terms of the total consumer biomass predicted with these different Earth system models. So this is, it goes from a minus 20, it's very small, minus 20 to almost no changes. And the figure to the right shows you the maps or the distributions of the total consumer biomass for the two contrasting Earth system models and the two contrasting RCPs. So GFDL 2.6, GFDL 8.5, and EPSL 2.6 and 8.5. You can start seeing, for example, that under, for example, GFDL 2.6, the projections of change or declines and increases in total consumer biomass are much lower than when we are using EPSL 8.5. That is the figure at the bottom to the left, where you really see there's some regions that are projected to suffer important declines in the biomass of marine organisms in the future. This is a comparison, as you have here, from 1999 versus 2090, 2099. Okay. Okay, and this study, basically this was published in PNAS, where we were not only using EcoOcean, where we were using 6 marine ecological models, and here you have the multimodal means or the ensemble models, where as in IPCC reports, we were averaging and looking at the uncertainty using these Earth system models, the RCPs, and the marine ecosystem models. Okay, so this is one of the works that we did under EcoOcean 1, but there's other studies that have been published and are available in the literature, where basically use or reanalyze the same results that I've showed you before. Here there's 3 papers from Andrea Brindum that she basically looked at more detail the changes of the projections of these 6 Earth system models that I presented to you before, one being EcoOcean, and also she zoomed in the North Atlantic Ocean, and also looking at what would be the change in this. I think she used the total consumer biomass in the different Canadian oceans, for example. There has also been some work by this BioRigs paper here by Íñigo Gomara, trying to relate some of the changes that we were seeing in Earth system models with teleconnections, or what's happening in terms of the environmental properties of one basin and what's happening in another basin, to see if there's really relations or these teleconnection, atmospheric teleconnections, that the oceanographers are very interested with. And there's also has been some work looking at the indicators that Fish-MIP was extracting from these models from the socioeconomic perspective. So there's this study by Danielle Boyce that links the outputs of the marine ecosystem models with the socioeconomic consequences that these predicted declines or increases may have in the different countries, in the different regions of the world. But I would like also to highlight here is that all the runs from these models, EcoOcean and other models that run at the global level, are accessible and in a free repository that is organized by the Institute of Climate Impact Research that you have here. Here you have the link where basically all the runs and all the standardized outputs are available for other people to download them and to further analyze them. And with that, the second part of my presentation, what I would like to do is to show you what we have done with EcoOcean when we participated in Fish-MIP, and then I told you that we decided to go back to EcoOcean and make it better and change or modify some things. This publication was out last year, and what I'm going to explain you today in this second part of the presentation basically is this publication. So if something is not clear, you can always go back here and look at the specificities in more detail. So what basically we have been busy for a while is we realized that we needed to improve the representation of species contributions in EcoOcean. We added more species to better represent marine biodiversity and the environmental sensitivities to these species. So in the first version, we had around 1,400 species explicitly represented, plus the generic groupings, functional groups that we were using. And in version 2, we have around 3,400 species explicitly represented. We also wanted to improve how EcoOcean responded to drivers and to changes in drivers. And for that, we did 4 things. We looked at how to initialize the distribution of the species in the models. And for that, we used native ranges. I'm going to explain to you how we did that. We also added something new. We basically created the cell-specific responses to environmental factors that I'm also going to try to explain to you how we did it. We also added a temperature-adjusted metabolic rates to modify growth and consumption per cell in the system, because in the first version we were assuming that the species were growing at the same pace everywhere, and we wanted to modify that. And we also updated the historical fishing effort, and we added a special management module in the new version of the global model. With that, we wanted to go back to EcoOcean and really test what would be the impact of these internal assumptions to add the native ranges, to add the cell-specific responses to our results, so the projections of our species distributions and biomasses. But also we wanted to understand the uncertainty in the drivers. And for example, if we use different Earth system models, Earth system model outputs, as I showed you before with Fish-MIP 1, or if we added fishing on top of climate, what were the main patterns? So this is the renewed or the complemented structure of the global model, and in gray, basically you have what we modified or what we complemented, and in the black, in these black stars, you see where was new to the model structure all the way. So you have the native ranges that were new to the global model. The Q10 that I'm going to explain was also new, and the management, the MPA management model that's also new to the global model. And we also use these cell-specific responses. Before getting into the details of the changes that we did to improve the model, I wanted to show you the groupings, because most of the time when we look at the trophodynamic model, we always show our functional groups, and I haven't done that until now for a reason, because I didn't want you to get stuck into which species we were including. Because here, basically, we have here around 50 or 52 groups. But these groups are very generic because they include lots of species per group. So some cases you can have 200 or 300 or 400 species, which represent the whole range of species that you may encounter in a group or that we have information for a group. What it's important to realize is that because it's a global model and it's special in every cell, later we subset the parameters and the we try to subset the parameters that are specific to those species that occur in that cell. So even, for example, if we are in the large reef fish, there's not going to be the same species of large reef fish in a cell in the tropics than in a cell in the Atlantic. Okay, so the species vary. What that doesn't vary is the groupings. And with that being said, let me try to explain to you what these native ranges were meant to do. So basically, in the habitat foraging capacity model, what we have here is for each species, the self-suitability is a product of three terms. It's if you see here with the little star, the red star, we have the input capacity, okay, which is the capacity that is obtained from external niche models. You can bring some information where you know where the species are. The second term can be the habitat capacity that is derived from the affinities of the habitat types that you have and the habitat distributions. That would be the second layer. And the third type is the niche capacity, which is derived from the combination of the response functions to the environmental drivers. And you can use a combination of the three or one or two. So at the beginning, we were using basically habitats. Then we introduced the environmental components or the environmental affinities with the niche model. You can also bring these priors, what we call the external niche models, and you can also totally override this and drive your distribution with, for example, a species distribution resolved with an external model. Okay, but when you use the internal capacities to calculate this cell suitability, the problem is that when you work at the global level, just using habitat affinities or environmental variables, it's very likely that you're going to overestimate or overrepresent where a species may be. And this is just because of numerical suitability. Basically, there's places where a species, it's suitable for the species to be there. So the model is going to start by predicting that those species will be there. But we know, because we have observations and we know the native ranges, that those species have never been there yet. So what we did to solve this challenge, that is not such a big challenge when you work at local or regional levels, because you are already constraining quite a lot of your view of the system and you know the species that are there. But when you work at the global level, new challenges arise. So what we did basically is that we limited the spatial distribution of each species groupings at the start of the EcoOcean run by using observed historical native ranges of the species within the functional groups. Okay, so to do that, we basically applied what we call the native ranges maps of species groupings, derived from species from the native ranges of a species at the beginning of the first time step. And then we allowed the habitat foraging capacity model to run and to allocate within those native ranges at the beginning of the first time step according to habitat affinities and according to environmental drivers. After the final first step, time step, we released that constraint. And then we allowed all the species groupings or the functional groups to move freely around the ocean. What they did basically is they distributed, obviously, according to their affinities in habitat or environmental conditions, some of them being able to move north, for example, if the temperatures were changing, or some of them was not moving at all. Okay, so that was one of the main changes in EcoOcean version 2. The second change was the fact that when we used the response functions for the habitat foraging capacity model, we realized that we had been too general in the way that we used the model so far, because basically the cell suitability is expressed as a group multiplier in the foraging arena, and it basically looks at the suitability of a cell for foraging, which then impacts growth and impacts production. What happens if you only use one function, one environmental function that represents the whole species groupings? You are being too general in the way that the species grouping can react to environment. Because as I told you before, we had, for a species grouping, we had a lot of species, some of them with probabilities to occur in one area and some of them not occurring in another. For example, tropical species will never occur in the tropics, in the polar systems, unless there's a lot of change in the future, and the other way around. So we ended up doing is using the species, so that the temperature responses for each species within a group to define the cell suitability per cell, per group, per time step. Okay, I think I know it's a little bit complicated to explain, but basically instead of having one response function per functional group, we basically had one response function per functional group per cell, per time step. So that increased the time, the computing time a lot, but also made us much more specific, or made EcoOcean much more capable to represent the local conditions of the groups in each cell. And it really has an impact in the way the species are distributed. The third big change that we did in EcoOcean was to, we realized that aerobic metabolic rates for species, they can vary from the optimal average temperature. For example, how a species grow and reproduce is linked on what are the environmental temperature. This is very important for ectotherms, so fishes, amphibians, reptiles, and invertebrates. So they are really dependent on the environmental temperature. And in the first version of the EcoOcean, we were for each functional group had an optimal growth and production in the same way in all the cells at the beginning, and that we wanted to change that. So basically what we did is to use what's called a Q10, which is a temperature coefficient. It's a factor to which a metabolic rate is adapted to or changes when the temperature is raised by 10%. So we explored the possibility to use this Q10 per cell, per functional group. In this case, we didn't go to the species level, but we went to the functional group to initialize EcoOcean, enabling one species, for example, the same species growing a bit different if they are in the tropics or they are in the subtropics, for example. Okay, and because there's a lot of literature in this Q10 coefficient, and a species, there's quite a lot of information even per species or per groups, we decided to try this concept and we applied it to fish. So all the fish have one Q10, and then all the invertebrates have another Q10. But the idea is to refine that and go a little bit farther in this analysis and see how that changes the results. The final update that we did in this version of the global model is to use a new version of the fishing effort database that Yannick Rousseau released in 2019. There is this publication in PNAS, and we used it to replace the historical fishing effort that we had in the first version. And we also allowed EcoOcean to represent the special special management options, like, for example, starting MPAs or closing MPAs as we go from 1950 all the way to the present, or even to allow us to do projections in the future. Okay, so with this, what we did is we went back, we implemented all this, and we used EcoOcean and as an environmental modeling platform, basically. So we wanted to test first the ecological hypothesis. So what was the impact of adding these native ranges? What was the impact of these cell-specific responses? What was the impact of these Q10 metabolic rates in our projections of understanding how the ocean may change in the future? Later, we went and we wanted to check what was the impact of having these alternative or alternative climate drivers. And later, we added to these climate drivers the fishing to see what was the impact of having fishing in the model. And we treat this really as an experimental platform. So you can turn on and off models, you can turn on and off drivers, and you can assess the impact of what you're seeing. And that's what we think it's a really interesting perspective that allow us to be so modular, and as Jeroen Stenbeck explained before. So I'm going to briefly walk you through some of the results and what we have seen by using EcoOcean and as an experimental platform. So the first experiment we have, what's the time? I am being, okay, I'm good. The first experiment was testing these 5 internal, what we call ecological hypotheses or internal configurations of our model. So we use, as you see here, experiment 1 to 5. We used the model with basically using depth and habitat affinities as the way to distribute the species. And in experiment 2, we implemented the cell-specific responses to temperature to see how that would impact our distribution of the species. In the third experiment, we implemented on the base experiment, we implemented the metabolic rates, the changes of metabolic rates based in temperature. In the fourth experiment, we looked at the native ranges, what was the impact of that. And in the third one, we combined all, as understanding there was a way to include climate change through different pathways in the model. We use one Earth system model, as you can see here, the GFDL model, because it's the one that predicts or projects less extreme changes in the ocean. And we use the historical and the RCP scenario 2.6, because as well, if you remember from what I presented at the beginning, is the one that it doesn't show so many changes in the environmental conditions of the oceans. Okay. We also looked, in terms of the outputs, we looked at the species groupings biomass, and then we looked at the ecological indicators in a similar way what we have done in Fish-MIP. So we looked at the total system biomass, total consumer biomass, and then biomass of commercial species, organisms that were larger than 10 centimeters and larger than 30 centimeters. As a way to synthesize and not get lost in all the potential information that we can get from this global model. Okay, so I'm going to show you for each experiment 3 types of results. The temporal changes, so what was projected from EcoOcean, the spatial changes, and some changes in terms of the species groupings. Here you can see some of the changes from the temporal domain. And basically what we saw here is that the alternative configurations or ecological hypotheses had moderate effects on aggregated ecological indicators. So when we were looking at the total system biomass or total consumer biomass, the differences or the turning on and off these specific ecological hypotheses that we had, if native ranges were important, or the cell-specific responses were important, had a moderate impact on our results. However, when we went into looking at the impact of indicators that focus on larger organisms, say the biomass of organisms that are larger than 10 centimeters or 30 centimeters, the results were starting to be a bit different. For here you have these 4 figures presented quite well. In some cases, we have a simulation, I think the simulation with all, that projects an increase of the biomass of organisms of 30 centimeters of height to increase by around 20%, while in the others the increase is lower. When we looked at the spatial changes, we started to notice some variability on the biomass trajectories under the different ecological configurations. You have all the figures in the paper, but basically summarizing, when we looked at the metabolic rates, we had a very important concentration of the biomass patterns in higher latitudes. And that's because of the impact of the temperature, differences in temperature in the global ocean. When we looked at the site-specific environmental responses or the native ranges, there was a wider overall distribution of the biomass in the ocean. When we looked at the base, so looking at depth, using depth and the habitat preferences as the base for distribution, we had specific areas where there was a lot of biomass, and then areas of the ocean where it was practically empty. And while when we used the 5X scenario, which combined all the ways to include this environmental sensitivity to environmental change, we observed this gradient of biomass increasing with latitude that we evaluated as a little bit more realistic to what we see in the ocean in reality, where there's lower biomass concentration in the tropics and larger biomass as you go up and go down to the polar areas. Also in this, you can see this in the first figure on the top. And in the second figure, basically what we did, we analyzed the changes in biomass for the different areas, and we already identified that all the models, this is an average, so this would be like an ensemble approach where we average the different experiments. We saw some declines in tropical and subtropical areas and positive increases of biomass in the high latitude areas as time passes and as climate change kicks in. In terms of the species changes, what was also quite interesting, we saw larger differences than for these more aggregated indicators. So it meant that it mattered what was our internal assumptions in terms of site-specific responses or native ranges or what we were testing for species groupings. And we also saw quite a lot of variability in the coastal areas. So you see this map to the left. This is for large reef fish, the confidence, the coefficient of variation of biomass in 2100. So you see that internally, what we put in our model makes quite an impact on what we are projecting for species groupings in terms of where they may be or how they are going to distribute in the future. Okay, and all the species, we also noticed that when we included the four components of adding this more realism into the way environmental drivers act in the ecosystem, we saw again these patterns of higher latitude biomass in higher biomass in higher latitudes as well. So we were quite pleased with that pattern that we that shows a more realistic patterns on what we observe in real life. Okay, then we went and we tested the contrasting climate change scenarios with how that would impact the ecological trajectories that we see of the model. For that, here in this table, you see that from this is experiment 6 to 9. As you can see, we used two Earth system models here because we wanted to have these two contrasting environmental data sets. So we used the two models that I showed you before, GFDL and EPSL. And we also used the two RCPs, the two most contrasting as well, 2.6 and 8.5. We wanted two Earth system models that are contrasting and also two emission scenarios that are contrasting as well. We used the internal configuration of the model that included all the all the modifications, internal modifications that I presented to you before. So the variability of the cell suitability, per functional group, and the temperature adjusted metabolic rates. Okay, and and that's important. And in these results that I'm going to show you, we assume there was no fishing. So we are playing in a non-fished ocean. Okay, so in general here, in terms of results for the temporal changes, we looked at the ecological indicators resulted in immoderate differences of temporal changes. We saw a little bit in the first part of my presentation. The Earth system models and the RCPs mattered in terms of what the projections of biomass of EcoOcean would give us. So you have it here in this figure as an example. We have on the top we have total biomass, total consumer biomass for the two Earth system models and the two contrasting scenarios or RCPs. We also saw that while GFDL was producing some positive or less severe negative impacts, EPSL was giving us very quite negative ecological results. So we saw a similar thing to what we had seen already in Fish-MIP results, that really these contrasting environmental drivers matter in the ecology, or what is predicted in the ecology. And the interesting thing is that it matters. So there is an amplification of this effect. So we see larger changes in larger organisms. So we have this effect of changes in environmental conditions that are increasing as the organisms are larger or are higher on the food web. In terms of the spatial changes, we saw quite a lot of spatial variability of the indicators as we had seen before, so that was no new. And we saw that the smaller changes or the predictions on how the ocean is going to be in the future change between GFDL, EPSL 2.6 and 8.5 being the most optimistic scenario when we use GFDL 2.6. Okay, that was also no new. But what was interesting to realize that here in this map, for example, you have the total consumer biomass prediction that is under EPSL RCP 8.5. And you can see that overall there's, and with the other scenarios as well, we predicting negative changes concentrated in coastal areas and temperate, tropical and subtropical areas, and some positive changes in northern regions and open sea areas. So as climate change kicks in, our model is predicting that changes in the tropical areas and subtropical areas may be mostly negative, while there may be some changes in the open ocean and some changes in the northern areas that may be positive. So there may be a positive effect of climate change in those areas. Remember that we are in a non-fished ocean. So we are in the experiment of, we are not fishing in the ocean in this experiment. When we looked at the species changes, we also saw that climate change impacted differently the species, and the larger those species were, for example, larger sharks were more impacted than the smaller or medium sharks. And smaller organisms even showed mixed results or some positive, in some cases, some positive impacts. This you can see here. In this figure, you see, sorry, the functional groups that we have in our model. They classify by if they are vertebrates that are not fish, so marine mammals, seabirds, and reptiles. If they are pelagic, so yeah, pelagic fish, demersal fish, or if they are invertebrates. And overall, we saw that the larger organisms, the larger vertebrates, no fish, they would all be predicted to decline under the two Earth system models and two RCPs, while there would be some changes, or the winners, what we call the winners, in small and medium organisms. And as we went from GFDL to EPSL, we saw some of the winners under climate change that would turn into losers. So it depends on which rate of climate change and which scenario you're in. Then some species show declines. So it's really not clear what's going to happen for them. What's also interesting as well is in a non-fish ocean, there are several species that seem to be winning or to be benefiting from climate change. Those would be the medium pelagic fishes, the bathypelagic medium fishes, bentopelagic small fishes, and the megabenthos in general. And again, we are in a non-fish scenario, okay? So the last experiment was to, okay, now we know our predictions and our uncertainties for the drivers of climate change. Now let's add fisheries to this. Let's see what happens when we add fishing. So with this, we added the experiment from 10 to 13. You have here in this table. And the only thing that is different is that in this case we had fishing. So we ran the same experiments of the non-fishing scenario, but with the historical fishing and until 2005, and then in 2005, when the historical data finished, then we assumed that the fishing effort would be constant in the ocean. Not especially, but the overall fishing effort would be maintained constant. Okay, so what we saw in terms of temporal changes was quite interesting. So we again saw that there's mostly a negative effect when we add climate and fishing. That was no surprise. So there's fishing aggravates the effect of climate. And we saw that both in the ecological indicators, so the more aggregated indicators, but also in the biomasses of many species groupings, especially of the large size ones. The large size ones, because of this trophic amplification, really suffer or are really projected to change negatively with these synergistic effects. So climate, when we added fishing on top of climate, we worsened some of the effects that we had seen with climate, and we dampened some of the positive ones. So some species or some indicators that were increasing slightly or notably under climate change would decline less or get to a negative trend under climate and fishing. Okay, and we saw the same the same pattern of EPSL RCP 8.5 being the most negative scenario that we could use. In terms of the special changes, there was also some interesting results here. So many of the areas that got worse under these cumulative impacts, tropical, coastal tropical and subtropical areas, were predicted to continue the declining in biomass of organisms. The Central Atlantic and the Mediterranean and the North and South Pacific, they all showed stronger negative trends under fishing. However, there are these areas again that show increases of biomass from a combined effect of climate and fishing. Those were the Arctic, the Antarctic, and the North Atlantic Ocean, especially under EPSL. So you see here these stars in blue, where these areas are that they go in the EPSL scenario, which is the most pessimistic ones. They would be the ones that we will be projecting to have a benefit or to show increases in some of the indicators. And in terms of the species changes, that was also quite interesting. Basically, what we saw is that the losers and the climate change would lose more. Basically, losers would be even losing more. The winners and the climate change overall would win less. So those species that are benefiting from a warming or changes in conditions would win a bit less. There would be also winners that then turn into losers, so then the fishing is hammering them and turning around the trajectory. And for me, the very interesting thing is that we identified two groups that would be winners. So they would win under climate change, and they were identified to still win or win a little bit more under climate change and fishing. And those were the bentopelagic small fish and cephalopods. And of course, these are big groups. So in these groups, there's many different species, some species that are much more resilient than others, but these are the general trends for the global ocean. So I thought it was an interesting result. So what we can learn from all of this and what we have done so far with EcoOcean. In terms of the ecological projections, we realized, or we confirmed, because we knew already, that the internal assumptions of what you add in your marine ecosystem model matters. And it probably matters more when you look at the specific species or specific species groupings. It matters a bit less when you're looking at the results at the general or more aggregated level, so total biomass or total consumer biomass, things like that. Environmental drivers also matter, and the uncertainty or the alternative environmental drivers from our system models or RCP have an important impact on the outputs of our ecological models, and that is translated into spatial variability and more pessimistic results or projections when you use the more pessimistic projections of climate change on the environmental conditions, which is quite logical. And the combination of the ecological mechanisms that we added to EcoOcean version 2 really projected some plausible ecological patterns. So we need to work much more into calibrating and validating our results, but we think it's quite a promise. Well, we are seeing promising patterns of how species and how the biomass distributes in the ocean. Under the non-fishing configuration, we saw the larger organisms show negative trends due to climate change, and the smaller organisms show mixed positive impacts. So there were clear losers and winners. When there's no fishing, it's easier to, I would say it's easier to know or to project what may happen to species. When there's fishing in the model, then things get complicated. Fishing intensifies the negative effects of climate change. In many places, it projects a stronger biomass declines for some species, or it dampens the negative, the positive effects of climate. So then you have confounding or effects or mixed effects that are, especially when you look at the local scale, are sometimes difficult to disentangle when you look from a local or regional perspective. And we also saw that several species groupings that were winning under climate change were losers under climate change and fishing, while others were projected to show positive results. So those would be the win-win. There's not many, but it was interesting to realize, for example, cephalopods were one of them, and in many places cephalopods have been proliferating or increasing in biomass, and that's of course, those are regional studies that show that, but it's interesting perspective to see that at the global level as well. For EcoOcean, for us, it's basically a baseline to further develop global marine ecosystem analysis, considering species, biodiversity, and how these species interact, how they interact in a food web. We think it's a great way to learn about the internal hypothesis and how we model the marine ecosystems and the global ocean in general, but also how external drivers impact the results that we are projecting to happen in the future. And the things that are going to come in this new version that we probably release hopefully next year, we are hoping to add new drivers of change. So not only sea bottom temperature and integrated sea temperature, but also the ice cover, the oxygen, and the salinity, which are probably fundamental to the dynamics of some of these species groupings that we were seeing. We are going to refine some of the ecological processes, especially in terms of the site-specific responses and the metabolic rates. And we are planning to work quite a bit in using this spatial management option that we have now to turn on and off marine protected areas and follow them. And our hope is that as EcoOcean matures and we understand more how to work with it and what kind of complexities we face in terms of data, and the data, amounts of data that we produce, that we consume are enormous. But as we go with it, we want to use it as for cumulative effect assessments of multiple stresses, and use it, for example, to test nature-based solutions to prevent, mitigate, and adapt to global environmental change. And in this direction, we already So I participated in this study, for example, the Campaign for Nature, where they were testing what would be the costs, the benefits, and the economic implications of protecting 30% of the ocean. And they have been running models and analyses both in land and in the ocean, and EcoOcean was one of the models that were used to look at what may happen in the ocean. And in this context, we are looking for projects and opportunities to use EcoOcean not only but also to look at what will be nature-based solution of conserving the ocean. But we would like to look at, for example, how it would interlink with restoring several habitats, or with combining it with a sustainable harvesting globally. What this may bring in terms of trajectories of change. And with that, I finish my presentation. It's quite a lot of material, but the good thing is that everything is written. Most of what we have done so far is written. So you have questions, please go ahead, and otherwise you can go back to the papers that are there and contact us. And thank you very much.
Thank you, Marta. That's a very comprehensive overview of some complex modeling that is necessary. We have time for questions, so let's open the floor.
I have a few questions. Hi, Marta.
Go ahead.
Thank you for that presentation. It is a lot of information, but I'm so interested in it, and I really hope to try to contribute to either Fish-MIP or EcoOcean with my own research eventually. So I'm trying to understand the structure of how everything is kind of working together. As far as the Earth system models, when you're using these, they still contain their own biogeochemical models, right? Right. So the turbulence and the wind and these other environmental factors, I guess, how do you account for, especially when it comes to lateral mixing, how do you account for that when it comes to nutrient or light limitation, and how that can be impacting biomass?
Well, that's a very good question. And actually, I can't answer it because I'm not an Earth system model. So what we do is we let them do what they are good at, and we just get their results. So basically, the outputs are our inputs. It would be eventually great to develop one full connected global ocean model that has all the way from climate to fisheries. But so far, what we are doing is we are interlinking models. So we are basically using the outputs of one model to put them as inputs in another model, and our outputs can then be used to socioeconomic analysis. But what you are asking is very relevant, and eventually in the future, it will be great to do that.
Yeah.
What we use is the primary production distribution. And we assume, our hypothesis is that all these mixing processes and all these processes that eventually bring to areas that are more or less productive in the oceans are being captured in that.
Right.
But we could potentially bring currents in. So for some regional studies, we bring currents and we mix the creatures or primary production in. But in this case, we haven't done that. We just use what they produced.
Okay. And as far as currents, you would add that to ecospace, right?
Yeah. And you could add them as special temporal layers where you have currents every month, and then every month things are moved because of the current. So you can do that.
Okay, cool, interesting. I have one additional question. When you were talking about Q10, the metabolic function, so does this change within each cell similar to temperature?
Yes, this changes. So basically, because you have a species that have an an optimal temperature, but they have a specific Q10. Well, here we don't work at the species level, so we work at the functional group level. So we don't go into as in the cell specific responses. We go to look for those responses per species. For the metabolic rates, we stay at the functional or at the grouping level, and then basically each species changes the production and the consumption in each cell because of the changes in temperature. Because we have this range of this range of temperature from the tropics to the poles that allow us to, when you apply the Q10, because we have different temperatures in each cell, the metabolic rates change in each cell.
Okay. Great. Yeah, that makes sense. Okay, thank you.
But there's still lots to do and lots to think about, because we could potentially do that per species or break down. We now only use it for fish, 1 Q10 for all the fish and 1 Q10 coefficient for invertebrates. We could go much deeper and use for different type of fish or invertebrates do that. But we wanted to see first if this would work and would give some realistic patterns. And we could, we are thinking on additional ways to make it better.
Okay. Okay, great. Well, I look forward to that.
Yeah, thanks.
Really, such amazing work, Marta. It's so ambitious. I think really important. I know it's really difficult to look at the species level, but I was wondering if there could be ways to look at species compositional changes within functional groups. So for instance, you might see a functional group, might behave a certain way in composite, but I would wonder if maybe some species might change within that group, and if there's any way to apply those preference functions individually within that group.
Well, that's what we did with the cell response function. Okay. What we did is we applied one response per species within the group. So those are species that were known to occur in that cell, where we used their response functions. And those are species that were in the functional group but were not known to occur in that specific cell. We did not use their response function to create the response function for the group. However, we have we don't know how the biomass of the individual species changes within the group. So the weighting that we gave to each response functions, each response functions to the species that occur in a cell was the same. If we would know their trajectories, or we would know their relative biomass, we could weight those responses with their biomass. Imagine we are in a cell where there's 10 species and one is 90% abundant, the other ones are 1%. Then we could do that and we would know how that changes.
Okay.
But we don't know yet.
Yeah, I would think it would be really cool, but of course, really hard.
Hi, I was asking about CIs, because you said that it was also included as a driver, and how is it?
CIs is not included in version 2, but it's going to be included in version 3. So we are now pondering with that. How it's going to, basically we're going to use the response functions of aquamaps, where basically have a response function related to the occurrence of ice or not. So whenever there is ice, that species has a response function or an optimal value that is, if that species is using ice, much higher than when there is no ice. And some species, for example, only occur in ice. Other species, when there is ice, they don't occur at all. We don't have that into account now, and that's one of the big limitations of our global model. But we are going to solve that soon.
Thank you.
Good. Any other? Ecotracer.
Ecotracer.
Yeah, I was just wondering if you have tried to use, or if you were thinking of using
That's a very good question. We have not used it, but there's definitely possibilities. And I don't know if Billy has tried to run the EcoOcean, the first version with EcoTrace, and I haven't. But it would definitely make a very cool project to do that.
Oh, definitely. I haven't tried it, no, but it's one of the things where we are very open to cooperation if anybody wants to do it. That's what it boils down to. That's how it works. One interesting thing in the work with Fish-MIP has been that it's a group of modelers coming together to do ensemble modeling. And when you do that, you really have to come into it with a cooperative mind. And part of that is that it kind of limits what analysis, what results we can be looking at, at least initially, to kind of a least common denominator. That we need to do things, we need to compare things that all the models can evaluate. And these models come with very different specifications, the way that they are specified. The way what's in them varies a lot. Some have really detailed analysis, the way down to a species level. That holds for William Chung's DBEM. It, to a large extent, holds for EcoSpace or EcoOcean as well, where we have a lot of information at the species level and also a really good handle of fisheries. Then there are other models that have hypothetical organisms in them that have characteristics, but you can't really identify them with anyone. That makes it much more difficult to talk about anything at types of fish, for instance, and some don't have fisheries. So it's this whole mixture that has to be accommodated in this. And one really important part of that is the experience that Marta was talking about, that to see what the implications are of including this kind of drivers and that kind of drivers and comparing that across models between different future scenarios, different models there. And that is so that's an interesting aspect really of being involved in these collaborative exercises such as Fish-MIP is. So something I would very much I must encourage you to look into if you are interested in global or regional modeling of climate change impacts.
If I can add to it, I think with this exercise that I showed you that we produced last year, but I think it was very interesting to realize is that the uncertainty in your results by bringing different drivers in terms of Earth system model outputs is the same that of the uncertainty that you obtain by changing the internal assumptions of your model. At least it's of the same order of magnitude, which means that you have to be open to turn on and off hypotheses in your models, and you have to be aware that the assumptions that you internally make in your models make a big difference on what you are projecting. So we are, the community is putting a lot of effort in Earth system models to have alternative or ensemble projections on what may happen in terms of temperature, salinity, oxygen, and we should be putting the same amount of energy in trying to understand what may happen, what these models project if we change the ecological hypothesis that on how we understand the ecosystem. So that's for me a very interesting output as well.
Yes. Yeah, I don't think we would have, if we hadn't been in Fish-MIP, I'm not sure we would run any experiments without fishing and see what the implications of that would be. We might not have been forced to do it, actually, because it's time consuming. And why on earth would you do that? Nobody's going to stop all fishing in the world ocean in next year. So it's still, these experiments are good for learning about model behavior. So that's a lesson from it.
Yeah, my question was, I think, Marta, you showed earlier in your slides, pH decreases with increasing temperatures, seawater temperatures, I guess. So would that be something you're considering and including in the EcoOcean 3 or maybe in future model potentially? Because I think it's pretty important if that's something that's going to be changing as well.
Yes, and I agree. I think it would be great. What it's basically stopping us is to have a good understanding of the response functions of the responses of the species to that. So globally, right? To have so we would have an understanding of the thresholds, or when things are going to start dying or being more vulnerable to predation, or then we could start bringing in those response functions and link it to changes in pH. So this is definitely something we can do, and I think as more research is being developed, we can probably include it in version 3 or maybe version 4.
So I guess there's not enough information models to input into the, or response functions rather.
I think there's quite a lot of information at the regional level or local level. I'm not sure. I'm not an expert in acidification of the ocean, but I'm not sure there's enough information to make it a generalization for specific groups like invertebrates or species that actually have calcareous covers, no, that they may be more affected. I'm not sure, but if there would be, and if you know if there is, that would be great to look at it together.
Yeah. Okay. Thank you.
If I can add to that, not all Earth system models provide pH yet, so that's another limitation. It was not considered in Fish-MIP yet.
Okay. Thank you very much.
But if we were to consider it, then of course including pH is just another environmental driver in the temporal spatial framework, and in EcoOcean, and for that reason in EcoSpace, it's just another environmental driver. So the key there is really what Marta talked about. What are the response functions? How do we handle that?
I guess related to this a little bit, I don't think I ever saw collinearity or how correlation is treated. Do we have to run any collinearity tests before we actually input any drivers into the system for EcoSpace or any of those modeling?
That's a very good question, because the more drivers you add, especially the way that the habitat foraging capacity model works, if they are important, you have to add them. But if they are redundant or they are highly correlated, what you're just doing is hammering down your species and the probability of a species to occur in certain places, because the foraging capacity model, what it does, it combines, so the cell suitability, it's a multiplication of the values of each environmental variable that you are including. So, of course, if you're in a cell where your temperature is good and your salinity is good, but your oxygen is bad, then the suitability of that cell is going to be bad. And if you add another driver that is bad, it's going to be much worse, right? Because you're multiplying it. So at the regional level, we have done some studies where we first check for collinearity of drivers, and even when you use species distribution models to first identify your environmental drivers that are meaningful to your species, and then also you can even extract the response functions from species distribution models, then that's a very good way to identify which are really significant, and then just bring in those that are significant.
Yeah, that makes a lot of sense, because I guess if you have what's significant, and if you know if two drivers are correlated, there's no reason to include both of them and just use the significant ones from other models.
Yeah.
Okay. just something I didn't think about before. And
what it's important to understand is that in the habitat foraging capacity model, the environmental drivers are multiplicative, but the habitat drivers is an addition. So you could be in mud and sand or sand, and that's not exclusive. So there's and that doesn't multiply on top of the environmental drivers.
Yep.
That's a little bit more forgiving. Yeah,
that very much answers my question. Thank you.
Sorry, I don't have the camera either. Thank you, Marta, for the speech. It was really interesting to learn about this global model. It's a simple question. I was wondering how is depth treated, because I think that in previous talks you said that you could separate different groups, but I was wondering if species are allowed to move within groups as an adaptation, as a response to temperature, or not, or if this is somehow accounted.
Yeah, depth is in this in EcoOcean is a layer. Is a habitat layer, and I think you can correct me. And you basically restrict. You can allow, or it's an environmental driver now. I'm not sure. Yeah, I'm not 100% sure. I think it's probably an environmental driver where you set response functions. So if you think depth is an important, so if you think that depth is really going to constrain your group, so it's not going to be able to move in a certain extent, you can use depth as a response with a response function. But if you want your species to really go deeper, you would probably have to exclude depth from your habitat capacity model. But Jeroen, you may have a better response.
Sure. Well, depth is the weird one in ecospace, and not only in EcoOcean, but also in ecospace. You can define various environmental drivers, but the one environmental driver you always get for free is depth, that you can add response functions to. So, but on top of depth, you can add several other ones. But you're right. I mean, the autocorrelation is a big issue. If temperature is a function of depth, then you might want to consider to make depth your temperature your main driver rather than depth, right? I mean, so you need to know your system well, and of course that's a challenge at the global scale. We talk about 3,400 species, etc.
Yeah, this brings another question, which is, do you have to test for collinearity at each single cell and species? So if you are adding different environmental layers, do you have to do that or not?
Perhaps we should, but it's an impossible job. No, we deliberately keep EcoOcean simple. If you go back to the figure of the paper, you see what we make the groups respond to, and it's really very coarse. It's the biggest suspects that we respond to, like just either temperature or depth. Nothing too refined, because often also the food web cascades take care of a lot, right? Availability of bioproductivity that swirls around the system that gets transported on the ocean currents, all that is delivered through the Earth system models. That already influences your food web dramatically. Then we use depth and temperature as proxies for other to explain other things that have been observing. Yeah, you can refine it, but how far do you go? I mean, how complete do you want to get? And are you going to see anything? I mean, you still see the trees from the forest. It's an interesting challenge.
Okay, thank you.
Yeah, I just to add here, remember that the depth preference functions we're using comes from aquamaps, and they are very wide. So they are representing where these species have been observed and a model output based on those observations. And as such, they really encompass quite a substantial range. Now, one more thing here is what is the interest? So in this model example we're talking about, what's the interesting thing about ecospace, EcoOcean? Rather, and to me that is the pedigree we come with when we come from EWE is a pretty good handle on food web effects and on fisheries effects. That's why we have our experience, and that's brought over in here. So I think EcoOcean gives one of the best handles we have on when species move because of climate change. What are the implications for, do predators and prey move together? Will there be enough food for species where they move to? Is there not production there? How primary production, how that channels up through the food web and provides limits for carrying capacity all the way up to the food web. Those are the things that we have a lot of experience with. So I think that's our strong contribution to the Fish-MIP ensemble and group that we come with that background.
Yeah,
and
if
related to this,
I can add that, Billy, it's interesting to realize that EcoOcean and some of the other models that indirectly include species interactions are the ones that are projecting changes of the future to be less dramatic. And that's because they allow species to how would you call it, in terms of to amortiguate, I don't know if amortiguar, to really hammer some of the negative impacts. Some species may go down, other species may go up, and that flexibility of the food web is what we capture in this with the trophic interactions. That is very interesting. Some species that don't have that, some models that don't have these species interactions are their projections are really dramatic on what may happen in the future, because everything may go down or everything may react the same way.
Yeah, so it actually looks like the food web effects here, the food web structure and what's in that may be adding some resilience to climate change. And that's, by the way, is something that is much less interesting when it comes to publications than coming out with predictions saying 50% decline and that kind of things. But that's a consequence of how things work here. But it's interesting. I was going to bring us to the question of fisheries, Marta, how fisheries are handled now that we have the new version of the fleet efforts. I think because it's not just a question of, we have fishing and we're going to be distributing that fishing on the map with a global fishing fleet. So what's the status now? Giona, Marta? Do you want to talk about that?
Well, so far we have been basically we have the global trends of fishing effort, and those trends are allocated. So we have them by country and by EZ. So basically we allocate those trends per EZ. So those are specially reallocated. So the trends are especially reallocated within a specific area where that country has been known to have fishing, and that's the status basically.
Yeah, that means we are actually working with a very large number of fishing fleets. For the first version of the model, it was by continent and or subcontinent actually, 15 cells for each, and we were dealing with more than 1,000 fishing fleets that we were allocating the projected effort from on the different scenarios actually. So it's complex, but it is not more complicated than what we're doing, what you're doing when you construct a model. It's just that by coding we found ways to not just do 10, but to scale it up and use databases to feed all these different models and only set out the rules for it. This relates actually to one of the foundations that Marta mentioned in her talk was the 66 large marine ecosystem models that we constructed back in around 2005 or so. That was part of developing a concept we call database-driven ecosystem model. Database-driven ecosystem models that, how do you make 66 models if you just think about it? That is, that's too many. You don't want to do 66 models. So what we did for that was actually to go into databases and construct all 66 models by sucking this data out of the databases, putting it into models, run the models, do tests on them, do more tests on them, trying to find ways of not having to look at 66 ecosystem models. And that really is the foundation for EcoOcean. That by doing it that way, the next step would be, because the 66 models were not spatialized, they were just put into 66 compartments, Ecosim models that filled the map. The next step, logical step, was let's try spatial. So again, the foundation there for that was let's do these things based on extracting things from databases.
Marta, I don't know if I said earlier, that was a really awesome presentation. Thank you so much. Actually, quite interesting and absolutely amazing work. So yeah, another question that popped to my mind, and I think I pretty much got it more or less answered with when you answered the question on depth. To implement plasticity response function for either temperature or anything else when it comes to a particular species. For example, like in my work on the species I've been working on, one of them is non-plastic whatsoever, and the other one has groups within the, if you want to call them different populations within the entire system, that some like completely different temperature range versus other ones. They're all cold water species supposedly, but some of them go up to 20 degrees and other ones prefer to go down and stay at 10 degrees temperature. So is there a way, I guess, to incorporate that, or is it more like what you mentioned with that, just a wider curve in the response function?
So so you mean there's a species, so different species or the same species with time?
The same species would have populations that are more adaptable, if you wish, than other populations of the same species in the same system. Like I work with lake trout, and they seem to have these different populations that do all kinds of different things. And I came across the other day a paper actually on I don't think it was coho salmon. It's one of the salmonids in the Pacific, and they found that one of the populations were more susceptible to higher temperatures than one of the other populations that actually was okay with the higher temperatures and seemed to compensate with different mechanisms, I guess, physiologically. But I guess, yeah, the question is, if they're more plastic and are able to compensate easier than others, is plasticity in general, is that a way, something you can implement into either ecospace or EcoOcean?
Yeah. Well, there's several ways, there's several ideas on how to do that. I think nobody has done it yet, but it's something that several people are thinking about that at the moment. And so there's for example, there's different ways. You could adapt their metabolic rates in specific regions. So imagine there's thought that when the species distribute at the edges of the distributions, they behave a bit different, or at the edges of the distributions, other organisms that are able to adapt to more extreme conditions. So you could make those cells, those species that are and individuals, populations that are in those cells with a slightly different metabolic rate, or you could change the cell-specific response functions now that we have a handle on how to change them per cell, and you could modify those response functions in those areas where you think that their population is behaving a bit different. So there's definitely ways to bring that in, and there's probably other ways to do it. I think the challenge for me here is to really know that is happening, and know a little bit to have the ecological data and the mechanism from what is doing to the species, and then you can model it.
Yeah, I guess compiling all that data for each different species or groups of species is it's quite a task.
But you could test it. You could test it if you have a model.
Right.
Sylvia's question reminds me of two things which I think are worth noting. One is that it's a big problem. I don't know what the right word for it is, if problem is the right word, but it's a question of downscaling. When you're working with global model, it's not straightforward to take such a model and just pick out a small area and look at that in detail. You don't expect a model, any model, when you downscale it, to be representative for a specific area. For that, you need to go back and take the first rule of modeling is, what's your research question, and construct the simplest model that can address that research question. And if that research question, like say here in BC, if this was something that related to salmon, we would definitely want to have the different types of salmon represented in that model. It would be a very different setup from the global model. So I would never use the global model for that. And generally, downscaling for all kind of models types that are involved, this is mainly something that's discussed in the context of climate change. For all of these models, downscaling is difficult and complicated, and something where the tendency is to construct regional models rather than downscale from a global model. This doesn't mean that the global model doesn't work with regards to its predictions for it, but when you downscale, you look at things with different resolution, and your questions are different, and again, the questions have to decide what model you use. It's not so that we construct a hammer and we go out looking for nails, but it is really that if these are the nails, we have to find a proper hammer to use for that. So that, I think, is it certainly is something that impacts climate change modeling a lot, to be aware of this. And you can also see it in Fish-MIP, where there are global models and regional models, some using the same modeling background. EWE is being used in both, of course, but some can only be used at one level. You need different models, basically, for different questions.
Well, part of the reason why I was asking this, and I mean, downscaling, yes, it is a problem for a global model, but if you're representing salmonids within the same group, then you have Chinook, which are very non-plastic species, and then you have lake trout or sockeye salmon, for example, that are much more adaptable as species. So that creates a problem right there with, and especially plasticity is very important when it comes to climate change, right? We're talking about changes that are going to affect the species in different ways. So that's part of the reason why I was asking this question. I understand it's important to try to separate the regional from the global.
And part of the assumption here is also that, like in the EcoOcean model, you won't even find salmon. You'll find them buried within a functional group with other things. And part of the assumption is that when you do that, when you take all of these species and you put them into a melting pot, a mixing pot, such as a functional group, that the average, when you put a lot of things together, you typically get something where the average isn't as impacted by the behavior of the individuals there. Like you take a lot of random numbers, put them together, bang, out comes beautiful normal distributions. And I think, Mimi, this relates very much to your question as well. And with regards to that, let me just mention that some of the papers that Marta referred to, the ones about Canada, for those papers, we actually took the output from the global model and used it at the Canadian level. But it's not downscaling, it's just extracting what's in that model for those regions. It's not going in, because when you go in, you want to look at it in more details. You want to be like saying, what's the difference between this side of the, you can't use it to go in and say, what's the difference between this side of this big area and the other side, like that's where we get into. We should probably
Billy, we
should develop a regional model.
Yeah, and we have a student presentation, right?
Only she's not here.
Oh, okay, because she was going to present one of those papers. Okay.
She was going to present one of those papers, yeah.
Okay, good.
Mimi, did you have something on this?
No, not really. I think that it's just more to what you're saying. It sounded like it made sense that obviously with your global model that it's useful for specific regional comparisons. And so that's the utility of it.
Yeah.
Not downscaling, as you said, but then you don't have to, I mean, it would be useful, I guess, to do that. And then if one could also construct specific regional models and then compare the results, then that would give some validity testing to the global model.
Yeah, let me add to it. There's because in Fish-MIP there's some areas where we have regional models. There's a paper being written by Tyler Eddy, and he's looking at what the regional models and the global models, not EcoOcean, but other like EcoOcean and other models are projecting for those areas, and he's comparing those. And what it's very clear is that these global models don't do a very good job in coastal areas. So you really want to have a regional model for coastal areas. And regional models do much better than the global model in those regions, of course.
The whole story starts with the driver data that, I mean, the global Earth system models don't capture coastal dynamics well, river runoff doesn't exist. It's just about the big gyres. And go ahead, Vili.
That was exactly my point too, that many of these models work at the 1 degree level, so you can't even see the coast, the ones that we are basing on, the climate hydrodynamic models. All right, but this being said, I still think it's absolutely valid from a global model to extract information at the regional scale, such as trends at that scale, but not to go in and look in more detail of what happens north of England versus east of England and that kind of questions. And the coastal zone is a big issue for these. It's one that the models are actually getting much better. The models that we're using for driving are getting much better at getting that coastal resolution into place. It took a while to get into their head that it was not just about the ocean circulation, but the productivity. Ocean productivity was coastal, and the coastal zones were really important. And that is a discussion that's been going on now for the last 2 decades. And during that time, there's been definite improvements in the output and the thinking of the people developing those global circulation models.
But Billy, just more to that point. I mean, so obviously you would not expect your global ocean model to reproduce accurately the absolute, I guess, what's actually happening, say, at a more regional level. But you definitely would expect it to reproduce the trends that you would get from those regional models. So that would be, I presume, an important thing to test, correct?
Yeah. We an example would be we use the model to estimate global biomasses of fish, but we have been very careful not to publish those, with one small exception, whereas the trend from those models is something we trust much more than those absolute numbers coming out of it. We have reached the time where we are going to close down this last guest lecture in Fish 501. And I would like to thank Marta and also that other person sitting there behind you for the work that you presented today. And I would like to thank you all for joining. And we will have a session on Tuesday morning here, at least Vancouver morning. It's going to be spring, actually. We might get over 10 degrees. It's really exciting. So on Tuesday, and we have no real program for Tuesday. It's going to be what you bring. So basically, think about how do I model, and we'll do that. And we'll talk a bit about the experience you have from this course. And what should we do next year? That would be one question I'm going to be asking on Tuesday for the course. And also, I would like to remind UBC and or registered student over, not just UBC. Well, you're all UBC students registered. That there is evaluation you absolutely must fill in, 100%. So thank you very much, all. It's snowing in Bergen. Ha ha ha. 10 degrees is fantastic in comparison. And we don't want to hear what the weather is in Barcelona. No, we don't want to. Thank you all. See you soon.